Hello. Good morning, everybody. I hope you guys are having just a wonderful day. I'm going to be presenting the first and the last bit of today's presentation, and Ricky's going to take the middle bit. So we're going to start off today's talk just giving everybody a bit of an intro to what lichens are, just for those of you that are less familiar with that. So lichens are considered a composite organism. They're a mutualistic symbiosis between fungi and algae that is just an amazing relationship where fungi are basically kind of farming the algae. The algae are the photosynthesizers and are the ones that are providing the primary productivity, and the fungi are providing protection and new habitat niches and all kinds of other benefits to the algae. The majority of lichens have their fungal partners being ascomycete fungi. You might see often little cup-shaped structures, which are their sexual reproduction structures on a number of lichens. There are a few that are other types of fungi, particularly a few basidiomycete fungi that do have lichen relationships. And interestingly, recent studies have shown that there's a basidiomycete yeast that is part of this symbiotic relationship, yet we have not very much information on what it's doing in there. So what I'm calling algae technically should be called the photobiont, and it is not just true algae that are present. A lot of lichens use green algae as the algal partner, but a lot of them are cyanobacteria, which aren't truly algae, although people call them blue-green algae. They are bacteria, so they're very, very different organisms. And some lichen relationships may even have both partners. So for example, the lichen that's shown there, that's Peltigera leucophlebia, and it has the relationship of both algal partners present. You can see the majority of the lichen being the light green, lime green color with the green algae, but then there's the little freckles, little dots on it, and those are actually little clusters of the cyanobacteria that are also present. Lichen are incredibly diverse in the number of species, as well as the different types of growth forms that they can have. Some, as we'll see in the next slide, have crust-like shapes. We call them crustose lichens. Some have a shrubby or goblet-like shape where they're round in cross-section, and we call those fruticose lichens. Others have a leafy look. We call those foliose lichens, and others form a lot of little scales. We call those squamulose lichens, and those can be subdivided further depending on how fussy you want to get. Lichens are incredibly important in our biodiversity in the province. We've got nearly 850 species recorded from Alberta. And with regards to conservation, they are incredibly important. According to the Alberta Conservation Information Management System, ACKNOWLDGEMENTS, 2/3 of the species of lichens that we have recorded from Alberta are rare or tracked species. So that means they are of conservation concern, though none of them have made it to the point of being a species that is being an endangered species or having any rank federally or provincially. So here's just a demonstration of the diversity of lichens that we may see in Alberta and across the world for that matter. So the top row there is crust-type lichens or crustose lichens in a number of different sizes and shapes. And in fact, the one on the top left is a species of Xylographa, which are one of my favorites because their majority of their structure is actually inside the substrate. The majority of it is inside the rotting wood, and we see other ones living inside of rocks or other kinds of soil. The next row there, verse 3, are examples of foliose lichens. We can see the first one being a Loberia species there with a big kind of funky looking depressions all over the surface of them. We'll be talking about some of those in our lichen transplant studies because they are a species of conservation concern in a number of areas around the world. And so those areas have received some attention for transplant work. They're also really important nitrogen fixers in in certain ecosystems. The next four ones are all ones that are kind of round in cross section and are what we would call the fruticose lichens. You can see some highly branched, some unbranched, and some forming kind of cool goblet shaped structures. So very diverse, and yeah, we could spend a lot of time looking at incredible diversity there. Okay, so lichens have a really important part of a lot of ecology. They are growing on top of substrates, but they don't have roots. They don't penetrate the substrate very much, if at all. They are attaching mostly just via the filaments of the fungal partner, and sometimes they form more complex structures called rhizines, which are used for attaching particularly to bumpier types substrates, and you'll see them often more on the leafy or the foliose lichens. And so that a lot of people have a bit of a misconception when they see an old dying tree and they see lots of lichens on it. They might think the lichens are hurting the tree or something like that, but that's not the case. It's just the fact that the lichens are utilizing those trees and those rocks and whatever as a substrate to grow on. And the reason you notice them more on the older dying trees is just because they have been there longer and they're very slow growing. And so, yeah, you'll see them growing on a lot of different substrates. A lot of lichens are growing on wood or bark or on rocks. Some grow on the humus and mineral soil. Some actually kind of grow on top of mosses and sometimes end up killing them, though they're not really parasitizing them. They're just kind of smothering them. And a lot of them are incredibly important to the ecosystems that they live in as being primary producers, photosynthesizing and adding to that productivity in the ecosystem. As I mentioned, some of them are important nitrogen fixers, and that's because they have that cyanobacteria in them. And so those cyanobacteria are able to do the nitrogen fixation. In rocky environments in particular, they help to create soil where there is no soil by secreting some acids into the rocks that they're growing on and helping to weather that rock, as well as just by their growth being able to die and add to the organic material there. And they are just incredible habitats for all kinds of little critters. If you ever pick up a lichen specimen, you'll just see all kinds of stuff crawling around in and around it. And so those little critters need those lichens for their home and for their protection and form the base of the food chain. So there are several types of communities where terrestrial lichens form the dominant ground cover. And so these are the ones that we are going to be talking about the most in this presentation because they're the ones that we're kind of focusing on, and you'll see why in a second here. So in areas such as very dry, sandy sites, often with jack pine, like we see in the bottom photograph there, we can see lichens forming a very dominant ground cover. And we'll also conversely see it in peatlands, particularly bog peatlands, where there's lots of light hitting the ground, and particularly also ones that have become raised higher than the ground level. These have, although there are still wetlands, they have a very dry surface, and that dry surface is a wonderful habitat for these lichens. And some species that we see as being dominant in these kinds of terrestrial lichen habitats are species of Cladonia, species of Stereocolon, or the snow lichen, species of Peltidra and Citraria, and we'll see some more on the next slide here. And so here are some of the dominant species of lichens that we see in these terrestrial habitats. And I'll just mention here that the first one here, Cladonia arbuscula / mitis, that's shown here is it's undergone some name changes in the recent history. So in most of the literature, you'll find it as Cladina mitis, but most are placing it within Cladonia arbuscula currently. I will probably go back and forth between them because I'm used to calling it Cladina mitis. So we've got that one as probably the most dominant one in both the bog type habitats as well as in the dry sandy habitats, often called the yellow or the green reindeer lichen. And sometimes people call them reindeer mosses, which is just terrible. Why would you call it something after another kingdom, for pity's sake? But anyways, after that, we've got Cladonia rangiferina, sometimes called the true reindeer lichen or the gray reindeer lichen, because it's got the one that's got the name of the reindeer in there, Rangifer. We've got Cladonia stellaris, sometimes called the boreal or the star reindeer lichen. And even though it is the next one here, Cladonia stygia or Cladonia stygia, is one that is, according to ACKNOWLDGEMENTS, a rare species in Alberta. I think it's largely due to people not noticing it, because I found it on almost every wetland bog reindeer lichen site that I've seen. There are some others that in some sites can be a big part of the community, species of Citraria or Iceland moss lichens, species of Flavo-Citraria as well, which are similar ones and sometimes called, I forget what they're called now, the common names. We've also got a number of other species of Cladonia that can form often very dominant parts of these communities. So if you look on the bottom line here, the Cladonia uncialis, Cladonia gracilis, we've also got Cladonia crispata, Cladonia multiformis that can all form dominant parts of this community at times, or at least co-dominant areas. And then we have the piltigeras. The piltigeras are an interesting one here because they're the only ones that are the leaf or foliose lichens. And another interesting thing about the piltigeras is that they are the nitrogen fixing ones. So we don't see them as being a big part in the diet studies that have been done on the caribou, but I think they are probably an important part of these communities because, as we may know, the nitrogen is a very limiting nutrient in terrestrial ecosystems, and these guys are fixing it. They're also very high in protein compared to the other lichens, so I wonder whether or not they might be a more important part of the caribou diet than we might give them credit for. And then we also have the stereocolon lichens, which are just fascinating ones, but you'll see them mostly on more disturbed areas. So lichens are facing a lot of threats, and we don't really recognize it very much yet. So they are very slow growing. A lot of them can grow only 1 millimeter or 2 per year. The Cladonia lichens, the reindeer ones, they have been shown to grow about 4 to 5 millimeters per year in Alberta. So that's still not a lot, but for lichens, that's actually a pretty, racehorse growth rate. And because of their slow growth rates and their relatively limited ability to disperse, they are quite slow to recover. And so studies that have been done after a fire disturbance have shown that they take, in the peatland type habitats, at least 40 years to start to recover to any level of significance, and in upland habitats much longer, 50 to 100 years, depending on habitat characteristics. In addition to that, they are also very pollution sensitive, and they are that way because they get most of their nutrients and their water not from what they're growing on, but from what they can absorb from the atmosphere around them. So from stuff that's dissolved in rainwater and dust particles, that is where they're getting the vast majority of their nutrients from. And so that means that being master absorbers from the air, that means they're also going to be absorbing pollutants out of the air. And so we'll see a very limited lichen diversity in cities and other areas where air pollution has become an issue. And so if you look at the bottom photo there, you can see this is a grid where they are quantifying lichens in order to do a biodiversity assessment and use them as bioindicators of air quality. So why is the ARCKP studying funding a study on lichens? Well, because they are a really key part of why we have caribou in the woodlands of Alberta. Woodland caribou, as we know, is a very important species of conservation concern throughout Canada and including here in Alberta, species at risk. And when the food is most limited for caribou, particularly in the winter, they are eating tons and tons of lichens. 1996 study found that 60 to 83% of the winter diet of the caribou is eating species of cladonia of some sort or another. And the only place where they can find that, the cladonia in quantities where we can assume that it would be worthwhile for them to dig all the way down to the snow to get them, would be in these communities, like we talked about, these pine communities and bog communities where the terrestrial lichens are dominant. And I think I'm turning it over to you here, Ricky.
Okay, thanks, Michael. So for the collection, so lichen are often collected in the spring or the fall to avoid extremely hot and cold temperatures. They can be collected using bare hands, using gloves, razors, knives, and sometimes rakes. It's suggested that lichen be collected moist because it's much easier to handle. So some have suggested to mist lichens prior to collection. So we found that timing collection after a rainfall event was quite effective because in the morning they were still quite moist and easy to handle. So it's recommended that people should not collect more than 20% of a population for conservation issues. So this is just the number that's most frequently cited. So when you've collected the lichen, one should clean it to prevent fungal infections and mold from developing. However, we found that it was quite time consuming, and other studies have as well. So we don't know how practical this is on a large scale. So these lichens, after collection, they're often stored in a dry and dark area. So they can be stored at room temperature, 4 degrees Celsius, or freezing temperatures, which is less common. So some studies have used down to minus 18. But in our study, we're using room temperature. It's also suggested to minimize storage time. So most frequently, lichens are used within days or weeks of collection. It's less common that they're used 1 year or 2 years after storage because it impacts the viability. So Denshin in 1988 studied lungwort lichen, which is shown on the right. So they clearly demonstrate an effect of storage time on lichen viability. So lichen were found to grow and increase in biomass after 22 and 90 days of storage. However, if the lichen were stored for 170 or 345 days, there were declines in lichen biomass, and they observed lower lichen viability and health. So clear impacts of storage time. So terrestrial lichens are ones that we can find growing along the ground. So not surprisingly, the more lichen that we put out, the higher cover we'll get. So in particular, reindeer lichen are a terrestrial lichen. So they can be spread as either fragments, which is shown in the photo on the left, or as a lichen mat, which is demonstrated in the photo on the right. But this lichen mat actually includes a diversity of lichens. Another example of a terrestrial lichen is crustose lichens. So these can be found growing on rocks along the ground. So what we can do is actually scrape the rocks and then take that lichen material and transplant it to the new sites. On the other hand, we can also just take the rocks and substrates that they're growing on and transplant that to the new site. So when transplanting these terrestrial lichens, we can use a variety of adhesives. So things that have been used are water, glue, synthetic resins, tree resins, but one should always be cautious about what you're adding to the lichens because they may potentially harm the lichens and their viability. So it was promising that in one study that water was quite effective for lichen attachment. So once the water dried, it facilitated attachment to the ground substrate. So arboreal lichens are lichens that grow on trees. So foliose lichens are often divided into segments and attached together using a monofilament string to form what are typically called lichen pendants. And then those pendants are hung onto trees. So examples of pendants are on the far left. So these pendants can also use hair lichens, which are found growing on trees as well. So what we can do is also staple lichen fragments directly onto tree bark. On the other hand, we can also we can attach those lichen fragments onto a substrate, including burlap, cheesecloth, gauze, and then we can staple that directly onto the tree. And instead of using lichen thallus, we can use their propagule. So we can use Ceridia in some cases. So in some studies, they have actually taken bark cores and then taking those bark samples and attached them to the bark of other trees, which was found to be effective. And also in other cases, people have created these mixtures that they painted onto trees. So what they did was they blended thallus fragments, jello, milk, and other fertilizer, and painted this directly onto the trees, which they found was actually quite effective. So a picture is shown on the far right, this technique. So when assessing lichens, we should take a variety of measurements. So we can take visual assessments, which are based on the amount of necrosis happening, discoloration, and bleaching. But we're told by some researchers, Sean Rapai and Brianna Collins, that just because the lichen look healthy doesn't mean that they're not stressed. So we should always utilize a variety of measurements, which would include biomass, size measurements, including length, width, height, and area measurements. We can take cover estimates in the field, and in the lab, we can take gas exchange measurements, which would include photosynthesis and respiration. So fluorescence has been utilized, which is an indicator of stress occurring in the lichens. So typically lower values would indicate higher stress in these lichens. And we can also collect chlorophyll content. So in the photos, we just have some of the methods that we're utilizing. So microscopes, calipers, scales, and quadrat cover analyses. So I'm going to be talking about 4 case studies that are quite relevant to our study that we're currently conducting. So there are well over 40 different studies that look at transplanting lichens. There's not enough time to talk about them all. So in this thesis, which was conducted by Sarah Duncan in Fort McMurray, Alberta, oil and gas activities had significantly fragmented the habitat of boreal woodland caribou. So a technique that we can do to restore caribou habitat is introduce lichen back into the environment, which should increase the amount of forage and hopefully caribou abundance without increasing the abundance of competitors like deer and moose, which would also attract wolf and other predators. So in this study, they investigated the effects of different substrates on lichen in both a young and an old forest. So in particular, it was a 12 versus a 24-year-old forest. So these authors had 3 different sites, or Sarah Duncan had 3 different sites where she laid out different plots or different blocks. And in these blocks, she had plots that were associated with one of 3 different substrates. So it was either bare mineral soil, moss, or leaf litter. And what she did was she collected Cladonia mitis fragments, which were 2 centimeters in length, and she laid out 25 of these fragments in each of the plots. And so what she assessed was the amount of growth occurring over 2 years. So she measured length, the degree of branching, and she also assessed for retention in these plots. So what she found was that the Cladonia mitis fragments grew an estimated 4 to 5 millimeters per year, which was actually in line with a lot of other research, which was quite promising. She observed apothecia formation in these fragments, which is actually not typically observed in this species, which was also an interesting finding for the study. And she found that there was higher retention in the younger forest on moss and leaf litter relative to the bare mineral soil, most likely because wind was quite significant in these younger forests, whereas in the pine or in the 24-year-old forest, she observed that there was no substrate effects, which was likely because the canopy was more closed and wind was less of an impact on the lichen fragments and their retention. So in the second case study, it was conducted by Ronalds and Grant. So large fires burned throughout British Columbia in 2014 in the summertime. So it had a significant impact on the habitat of the woodland caribou, specifically the southern mountain caribou population. So what these authors did was they went to test various transplant techniques, including manual versus aerial deployment. So this was actually one of the first studies to deploy lichen using an aerial method. So the authors had 2 different sites, and they laid out different transects along these sites, which were 100 meters in length. They set up monitoring plots at these transects to assess for the amount of lichen coverage. So they collected Cladonia mitis, Cladonia rangifera, uncialis, and Stereocolon tomentosum fragments, and then they fragmented them using their bare hands, which is why there's a bear in the photo, or weed whacker. And then they deployed these they deployed the lichen fragments using their hands or using a leaf blower into the plots. The other technique was to deploy the lichen using a helicopter. So in the end, they found that the leaf blowers were not effective because they typically got clogged with lichen material. So they often used their hands. And lichen coverage didn't differ significantly between the aerial method and the manual method, though it was much more effective to deploy the lichen aerially, though it was much more expensive and costly to rent the helicopters and have the staff for that. So for the third case study, it was conducted by Sean Rapai and colleagues. So in northern British Columbia, there was fires in the summer of 2014 as well, but this impacted the northern mountain caribou population. So in this study, they aimed to test different transplant techniques, specifically looking at the deployment of fragments versus lichen mats. So in this study, they had 100-meter squared plots where they added 100 liters of lichen that were either that consisted of either fragments or lichen mats, or both lichen mats and fragments. The control had no lichen applied to the plots. So in this study, they utilized Cladonia mitis, Stygia, stellaris, and uncialis. And what they found was that it was much more effective to, or much more time consuming to deploy the lichen mats opposed to the fragments. And this study is currently collecting data, yearly measurements on the lichen, and is actually one of the sites that we hope to visit in the near future to assess the historical sites. So for the fourth case study, it was conducted by Rotorier in 2007 at the Vindeln Experimental Forest in Sweden. So forestry and overgrazing have been a significant problem in Sweden. They have reduced the biomass of lichen that is available for Eurasian tundra reindeer, which is a subspecies of caribou. So what they wanted to do was actually test various substrate effects on lichen growth in both a cut block and an old growth pine forest. And they wanted to assess differences in fragment size. So in this study, they had 1 by 1 meter quadrats laid out with 30 individual Cladonia mitis fragments. So what they did was they tied each of the fragments with string to be able to follow their retention in the plots. So these fragments were either 1 or 3 centimeters in length, and the substrates were either mineral soil, moss, twigs, or bark, so 1 of 4 different substrates. And they aimed to assess for the retention of the fragments in the plots and also the recolonization from outside the plots as well. So they found that for the 3 centimeter fragments, there was higher growth rates observed relative to the 1 centimeter fragments, likely because they had more access to light because they were much larger. And these 3 centimeter fragments were more likely to be retained within a plot because they had greater wet weight. In the clear cut, attachment was higher on moss and bark, likely because wind was more of a significant factor in these killer cuts, whereas in the pine forest, there was no differences in the substrate. So these are kind of similar to the results that were found by Sarah Duncan, where the effects of substrate were more significant in the younger forest versus the older forest. So in their plots, they found that mineral soil was actually good for colonization from outside of the plot, likely because there was less competition within each of the plots to begin with. So Michael will now cover some operational scale details.
All right, thank you. So in thinking about how we could potentially take this onto an operational scale, we're going to be doing a lot of work to flesh this out in our project. But when it comes to collection of lichens, the studies vary. There haven't been very many studies that have looked at optimizing collection time, but this is kind of like how long it took us. So in the Rapai study, they found that when lichen was sparse, it took them about 30 minutes to collect 100 liters. When lichen was abundant, it took them about 90 minutes to collect that. Actually, I think that should be the other way around, shouldn't it? Yeah, I think we have that the wrong way there.
Yeah, other way around.
Yeah. Sorry. And then the Ronalds and Grant study that we were talking about took them about an hour to collect about the same amount, 100 to 150 liters. In our study, we weren't focusing as much on speed of collection. We did a lot of ecosystem studies and percent cover of both the lichens species that were present as well as all the plants that were present. So we did 10 1 meter by 1 meter quadrants in 2 days and 8 quadrants in one day when we were focusing less on ID and just getting on the lichens. As far as how long it takes to transplant them, the RAPI study, which they were just using their hands, and they were finding that it took them about 5 minutes to spread fragments on a 10 by 10 meter plot, and to actually take chunks and kind of dig them in and put the mats in took them about 10 to 15 minutes per plot. In the Ronalds and Grant study, when they were dispersing it manually, took them about 2 hours to do a 2,000 square meter transect. But when they did it through the aerial dispersal, it only took about 15 minutes to do the same. But as Ricky had mentioned, it was a lot less cost effective. All right, so yeah, so as far as the costs are concerned, Routier, their study found that the average cost for the manual dispersal was about $650 per hectare when they are going for the lower cover, and about $3,200 when they are going for the higher densities of lichen. In the Grant and Ronald study, they found when they were doing it manually, it cost them about $350 per hectare, but when they did it through the helicopter, it cost them about $125,000 a week for all of the crew and accommodations and fuel and helicopter costs. So then this takes us to where we are at as far as trying to figure out what are the gaps in knowledge that we have to try to take this lichen transplantation and turn it into something that is an operational scale that would be a cost-effective thing for us to be able to do in forestry as well as in other industries. So some of the challenges that we have is just we don't really know too much about how to ideally collect and disperse the lichens on the landscape. The information that Ricky had done or presented is mostly people saying what they did, not testing out different techniques to see what's the ideal technique. It's kind of like, this is what we did. It worked or it didn't. Another challenge is understanding the timing of collection and application. When is the best time to do that? And making sure that we respect storage times. And again, the storage information that we have, there's only the one study that really looked at how best to store them. And so it would be good to know what are the ideal storage conditions. The growth conditions for the lichens, of course, are another significant issue, especially when we are wanting to try out different techniques within the greenhouse situation where we can have a little bit more control. Knowing the ideal temperature, light, and watering conditions are going to be very important for us to be able to study them in that kind of a greenhouse setting, but also understanding for field applications when would be the best times for those to happen based on and best times and locations for application of these techniques.
I think we found that there's very few greenhouse studies, so
yeah,
further work should be done on that.
Yeah. And so again, the slides that we just showed a couple slides back looked at some cost effectiveness of lichen deployment, but yeah, trying out different techniques, trying out different timings and applications are really, that kind of work is just in its infancy. So scaling it up to large scale operations is kind of a key thing for us to make this something that is applicable. And one of the key things that a lot of studies have shown is that whenever we have a more open area, these lightweight fragments tend to just blow away and only kind of get caught in certain areas, which kind of minimizes how well they are dispersing on the landscape. Another important thing that is a key thing that we're going to be looking at is determining what the ideal size is for the fragments, particularly looking at that on different substrates because it's likely to differ from one substrate to the next. We want to be able to figure that out and kind of determine the best sizes so that we make best use of collected lichen material and we're not just stealing from one to add to another and just causing as much damage as we are doing for restoration. So we want to be able to minimize the disturbances and spread that over the largest amount of area that it can cover and provide good results. Additional knowledge gaps is just where the studies have been done. We know that different areas are likely to have different impacts due to climate. So as you look at the number of studies that have been done, there's only one that's been done in Alberta, that Sarah Duncan study. Most of the other ones have been done in BC, US, or a number of them done internationally in Sweden and Poland and similar locations. And very few of these have been done with terrestrial lichens. So you'll see a lot of the other ones have been done with fruticose or foliose lichens or with crustose lichens, or and so kind of limited applicability there, kind of like looking at bird studies to see how well moving caribou would work. It doesn't, probably has a limited applicability. The other big thing is long-term studies, looking at whether or not this lichen transplantation is actually going to take in the long term. The majority of the studies have been just 1 to 3 years, and so with such a slow-growing group of organisms, it's tough to say whether or not those plots would actually maintain those lichens into the future. And other significant gaps in the research is just a lack of the research on other lichen species that are important components of these lichen, terrestrial lichen communities. There are other species besides the species that have been studied in a lot of these projects that are fed on by caribou, and then yet other species have not been included, yet may be very important part ecologically of the lichen communities. And I'm kind of partial to the Peltigera hypothesis that they are probably important parts of these communities and yet haven't been included in any of these works. So yeah, we haven't seen any work or minimal work done on Citraria or Stereocolon. Most of the work is focused just on the reindeer lichen members of Cladonia, and yet there are so many other Cladonias out there as well. So we just want to acknowledge our sponsors and our partners here. As it was presented at the beginning of our talk here, our funding is through the ARCKP, which is a branch of the FRI. And of course, Portage College, where I'm from, is helping to sponsor my time in this research project. And Nate is got a number of members also assisting and providing resources for the project.